Now that you've survived Chapter 3, which is a huge chapter that really sets the foundation for the rest of the microbiology course, we're ready to move into Chapter 4. Now, this is a much shorter chapter, but it requires some of that dreaded memorization that students are not always really excited about. In Chapter 4, your job is to correctly classify different groups of prokaryotes while really appreciating the diversity among this group of organisms. Now remember, our prokaryotes are going to include our archaea and our bacteria. The prokaryotes are going to be the organisms that do not have membrane-bound organelles. So as we move through this chapter, the first term we need to make sure we understand is ubiquitous. Ubiquitous tells us that prokaryotes are found everywhere. This is going to relate to the lab that you're going to work on, your first actual wet lab you'll use your hands and lab kit for. You're going to swab some different spaces around your house, apartment, wherever you live, and see just what's really growing there that you can't see with the naked eye. As we move along in the chapter, there's a few terms that you need to become familiar with. The first term is community. A community is the term we use for how the prokaryotes live. A community is going to be a group of interacting populations of organisms. A population is a group of organisms that belong to the same species and are in a limited geographic region. When we think about a population, those organisms that are living in that close geographic region are going to typically have some sort of effect on each other. And the type of relationship and type of effects we see, we call different symbiotic relationships. And this table here, directly from your textbook, shows you some different types of relationships. With a mutualistic relationships, both populations within a both groups within a population are going to benefit from each other. With amensal relationships, one group will be harmed while the other is unaffected. With a commensal relationship, one organism will benefit while the other is unaffected. With neutral relationships, both organisms can live together but really have no effect on each other. With parasitism, one will benefit while the other is harmed. Most of the relationships we'll study in this particular course will be mutual relationships and parasitic relationships. With the mutual relationship, we think about something like the bacteria that lives within our gut. The bacteria has a nice environment full of food that we constantly provide the bacteria, and then that bacteria provides us some benefit as well, helps to produce certain vitamins in our stomach, all sorts of different things for us. Parasitic relationships are going to be relationships where the bacteria is typically going to harm us as a human, causing an infection. Your next term to memorize is a microbiome. The microbiome is going to be made of all prokaryotic and eukaryotic microbes that are associated with a certain organism. If we think about our human microbiome, there are millions of different bacteria and eukaryotic microbes all living on us and in us at a time. This is not an image that you need to memorize by any means, but I just want you to start to get an appreciation for the amount of organisms that we have living on us and in us at all times. We now move into the part of this chapter where you're going to need to start to memorize and classify. So the main way we separate our groups of prokaryotes is whether the bacteria is gram-positive or gram-negative. So you can see the first set we'll go through are the gram-negatives. Remember, gram-negative bacteria are bacteria that have the thinner peptidoglycan cell wall with the additional lipoprotein fatty layer on the outside. I would highly recommend, as you start going through these, you may want to create you a table to help you study with, whatever really works best for you. I've separated this out into the PowerPoint, but it may be helpful for you to write this in some different ways. So as we go through our gram-negatives, our first group are the proteobacteria. Within the proteobacteria, we have the alpha group. The alpha group, again, of course, are all gram-negative and are capable of living in low nutrient environments. So there's a table here showing you quite a few of the alpha proteobacteria, but I've tried to highlight on each slide some of the important ones. So the chlamydias are important. We'll study those as we go through STDs. And then the rickettsias are going to be important as well. These cause things like typhus, Rocky Mountain spotted fever. So we'll study those as we go through the course. So our first gram-negatives, the proteobacteria, we have alpha proteobacteria, and then we have beta proteobacteria. The beta proteobacteria are going to all be in the gram-negative category, and a couple important ones are going to be the Bordetellas and the Neisseриas. Bordetella is a genus of bacteria that causes the nasty whooping cough. We'll study that quite a bit. And then the Neisseria group is the genus that is the causative agent of gonorrhea, which we'll study in STDs, as well as one form of meningitis that we'll study as we go through neurological diseases. Staying in proteobacteria are gram-negatives, so we then have the gamma proteobacteria. Very diverse group within the gamma proteobacteria. A couple of important ones you should recognize right away: Escherichia, that's the genus for E. coli. And then we have quite a few other bacterial gastrointestinal causative agents such as Enterobacter, Legionella, Proteus, so quite a few that we'll study. Vibrio causes cholera. The next group of proteobacteria are the delta proteobacteria. These, continuing in the gram-negative, these are going to be non-disease-causing bacteria. They are going to be more sulfate-reducing bacteria. Not going to really study those quite as much in the class. And then our last group of proteobacteria are the epsilon proteobacteria, with two important groups, Campylobacter and Helicobacter. Campylobacter is one of the main causative agents of human gastrointestinal upset. So you've always heard you shouldn't eat raw chicken. The Campylobacter is your reason for that. And then Helicobacter is the common bacteria that we see in a lot of peptic ulcers as well as stomach cancer. Staying in our gram-negative group, we have the non-proteobacteria. This group is going to house the spirochetes. Spirochetes are really highly twisted bacteria that have this axial filament that wraps around. Axial filament is almost like a flagella that wraps around the entire length of the bacteria, and it can help it almost move like a corkscrew. It can tighten up really tight. It can spread out. They really uses axial filament to move around a lot. A couple of important groups within the non-proteobacter would be Treponema. This is the causative agent of syphilis. We'll talk about that with our STDs. And then there's some free-living ones that live in water and other groups. Continuing in our gram-negatives, we see our phototrophic bacteria. These are bacteria that are capable of photosynthesis. Photosynthetic bacteria are important to us because they can produce oxygen and really provide food for themselves, just like the plants do for us. Now let's take a quick look at the gram-positive bacteria. The gram-positive bacteria are separated into two groups. First, we have our high GC content, and then we'll have the low GC content. You may recall from your biology class that there are 4 different types of bases that make up our DNA. We have adenine and thymine, and then we have guanine and cytosine. The more guanine and cytosine, the high GC content, means you have a more tightly hydrogen bonded DNA. So that's just kind of how they're classified. Within our high GC content, we have the actinomyces group. They are going to cause a lot of mouth infection, so we'll study those, especially for my pre-dental hygiene majors. And then we have the Mycobacterium. Mycobacterium are interesting. They have a special type of waxy coating on the outside to help them live in dry environments, but it makes them harder for us to kill with antibiotics. So we'll study Mycobacterium when we study diseases like tuberculosis and leprosy. And then we have a few other groups within the Hyg constant gram-positive bacteria. Corynebacterium is the genus for the causative agent of diphtheria. And then we'll study a few others as we go along. The low GC content are going to contain groups like Clostridium. Clostridium is a spore former, causes gangrene, tetanus, botulism. Study those quite a bit. Bacilli is the other group of our spore formers, causing things like anthrax, MRSA, toxic shock syndrome, lots of different diseases. And then the last group that you need to just become slightly familiar with in this chapter are the Archaea. Archaea are prokaryotic organisms that are unicellular, just like your other prokaryotes. The difference is their cell walls do not contain peptidoglycan. The archaea are going to be found in lots of environments. Some of them will grow at normal temperature ranges, but the really neat archaea are the ones that can grow in extreme environments. They can grow in extremely hot environments like boiling lava, very cold environments like the Antarctic. They can live in very high salt content, just really remarkable habitats where these prokaryotes can grow. And then this is your 5 major phyla that you just need to recognize the name and understand they belong to your Archaea group. So as you can see, that's a much shorter PowerPoint, a much shorter lecture. So I highly recommend you sit down and figure out a way to help you classify these different prokaryotes and get an appreciation and understanding for the diversity.